Good morning, everyone. Thank you so much for joining us. We are about to get started on the first of this series of webinars that is the Kernza meeting. I'm going to share my screen just for a minute to give a couple of housekeeping details. And let's see. Okay. So this webinar is going to run about 2 hours. The attendees, you will all be muted throughout the webinar. If you have a question, you may be invited to share it with your voice, but the easiest way to do that will be through the question and answer function. And a moderator will be monitoring all the questions and will either read your question or invite you to ask that question with your voice. In order to submit a question, use this QA function here at the bottom of the Zoom window, and a separate window will open where you can type your question, and your content will appear to the panelists and moderators, and we'll be able to read them. So if you submit them in the chat function, which is here, we may not see them. So please submit them through the Q&A function. Thank you so much for joining us. And with that, we will get started.
All right. So Tessa, is everyone seeing all the panelists at the moment?
Yes.
Okay. So good morning, Lee DeHaan. I'm the lead the Kernza breeding program at the Land Institute. I've been organizing meetings sort of like this. This is the 5th year going now. It's a big change from how we've done it in the past, but our planned meeting here in Salina had to be canceled, and so we put this together quickly. We hope it's in some ways even better and more exciting to everyone, because we've been able to involve a lot of people from other countries that we otherwise would not be able to participate in this meeting. So I'm really looking forward to the next 7 days of 2-hour sessions. I hope you can join us repeatedly as we start each day at 8 o'clock in Central Daylight Time here in the United States. Thank you for those of you on the other side of the world who might not be the best time for you. We hope you can join us, or we're going to be trying to record these and get permission from the speakers to post them also as recorded videos. So if the time doesn't work for you well, hopefully you'll be able to see these online at a later date. With that, I think we're going to get started. Our first speaker today is Kayla Altendorf from the University, formerly from the University of Minnesota. Kayla recently graduated from there, where she received her PhD with Jim Anderson. She's now a research geneticist with USDA ARS in Washington State. She's studying hops there. Today, Kayla will present some of her PhD research on yield components in intermediate wheatgrass space plants and the genetics of domestication traits. So Kayla, we're looking forward to hearing what you have to tell us. Go ahead and get started.
All right. Thanks for the introduction, Lee. And thanks so much for everyone for joining. I'm looking forward to sharing with you a little bit about some of the work I did during my PhD at the University of Minnesota. Can you guys hear me okay? We're good.
Perfect.
Okay, thanks. Okay, so the title that Lee assigned me was Yield Components and Nested Association Mapping, which I thought was spot on. So here we go. So there's a lot of excitement around Kernza. And everyone's looking for ways to get from our breeding program to a cultivar release more efficiently and more precisely. And 2 ways to do that are to implement genomics assisted breeding, as well as improving our understanding of our basic traits of interest. And as many of you know, yield is a major target of selection in intermediate wheatgrass, but yield is a really complex trait. Yield is influenced by the number of reproductive tillers that a plant or a stand produces. It's also influenced by the number of spikelets and florets that a single one of those reproductive tillers produces. And then on top of that, There's variation for the number of seeds or the number of floret sites that are actually utilized and the size of the seeds. And another layer of complexity is that we evaluate intermediate wheatgrass parents in a space plant environment. But as many of you know, we actually grow Kernza in a sward environment. So these two different environments are obviously very different with very different levels of competition. And we know relatively little about yield components in either one. And today I'm going to focus on trying to improve our understanding of what's happening in the space plant environment. And when breeders are looking at intermediate wheatgrass in the space plant environment, there are two different ways that they can evaluate yield. The first is looking at yield on a per spike basis. So this includes harvesting 10 individual spikes from each space plant and evaluating yield by dividing the yield by 10 to get the unit, or to get yield per spike unit. Another way to evaluate yield is to harvest all the spikes from a space plant and evaluate yield on a per plant basis. And if you're a breeder, one really simple way to try to determine whether there are other traits that you might be able to select on that would be positively correlated with yield is to run a simple correlation analysis. And I'm showing just 1 year of data from St. Paul in 2017 with my space plants. And I'm going to show yield per plant and yield per spike. So those two different methods of evaluating yield correlated to a whole series of yield component traits. So we have things like number of reproductive tillers, how soon the spike emerges from the boot, some various biomass traits, including flag, leaf area, height, stem diameter, as well as the number of opportunities that a spike has to produce seeds. So things like spikelet density, florets, spikelets, the weight of the seed, and how many floret sites are utilized. And this is a Pearson correlation table. So numbers that are bolded are significant, and I use conditional formatting here. So higher numbers or a higher correlation are darker green. So you can see that a lot of traits are positively and significantly correlated with either measure of yield. And it's kind of hard to interpret as a breeder. So which of these are truly important if so many of them are so high? And a seminal paper in 1959 looking in crusted wheatgrass seed production developed this method for trying to simplify this correlation matrix. And they developed a method known as, or they utilized a method known as correlation path analysis in the context of yield components. And using this approach, I'm just simplifying and boiling this down to a few yield components looking at yield per spike. So they were able, using this method, to assess the direct effect that various yield components have on yield. And in addition to that, they were able to parse out which or what effect these varying yield components have on each other and have on yield. So you might have remembered from the previous table I showed that height was really positively and significantly correlated with yield. But using this approach, we're able to parse out which of that effect is direct and which of that effect is associated with other yield components. So what effect yield or height has on spikelet density. So this is a really powerful approach that allows us to paint a clearer picture of what's going on with yield components. And a more modern method of this is called structural equation modeling. And this was first applied to yield components in another seminal paper in 2011. And so I chose to apply structural equation modeling to my data. And so to do this, you have to develop a model in advance. And so I used the literature on wheatgrass and other small grains crops to paint a picture of how I thought these yield components might be interacting. So I determined that yield on a per plant basis is influenced by the yield on a per spike basis times the number of reproductive tillers. And yield on a per spike basis is determined by the weight of each seed, how many are utilized, and then how many opportunities there are to produce seeds. And then I tested whether a whole variety of biomass traits might be playing a role, as well as maturity. And I'm showing the results here of the structural equation modeling. So on the y-axis, I have all the yield component traits. And in each panel, on the left panel, I have yield on a per plant basis, or YPP, and yield on a per spike basis. And traits that don't have a bar were deemed insignificant by the structural equation modeling method. So those are excluded. And the darker blue is the direct effect on each type of yield, and the light blue is the indirect effect. And this is just for St. Paul in 2017. So you can see that when yield is measured on a per plant basis, reproductive tiller number has the greatest effect on yield per plant, followed by yield per spike. However, when yield is measured on a per spike basis, floret site utilization is the greatest contributor to yield, along with varying yield components like 1,000 grain weight and opportunities to produce seeds. And this trend holds up in St. Paul in the second year, where reproductive tiller number again is the most important component on yield per plant, and floret site utilization and varying yield opportunities for florets and spikelets are closely followed behind. And this trend also holds up in a different location down at the Land Institute. And you can see again that floret site utilization is the most important and reproductive tiller number. So from this model, we determined that although some biomass traits appeared really positively correlated with yield, that their effects were fairly minor. And you can see in the case of height, it does offer a positive benefit, but it's not enough that would indicate that we should be selecting for height, not that we want super tall plants anyway. Flowering time played a minor role in it was not significant in the first year, but in the second year provided a small benefit at St. Paul, and then at the Land Institute, when there was a severe drought, it provided a pretty substantial benefit. Earlier flowering led to greater yield. So in the end, we show that when yield is measured on a per plant basis, reproductive tiller number is probably under, and when we're selecting on yield per plant, we're probably strongly selecting on reproductive tiller number. And when we're selecting on yield per spike, we're probably strongly selecting on yield component traits and floret site utilization. And there's some evidence that recently came out with a paper by Mitch Hunter et al. That show that one of the components of yield in a sward environment was that yield, the ratio of reproductive to all tillers declined with stand age. And we haven't, as far as I am aware of, determined that whether reproductive genotypes that reproduce or produce a lot of reproductive tillers, whether that holds up in a spaceline or in a sward environment, and whether that's something we want to select for. So breeders should make this consideration when determining how they select for yield. So looking at, oh, sorry, I'm a little out of order here. Another thing I want to mention is that this was a very comprehensive study of yield components over multiple years and multiple locations. So I wanted to just look at location averages. And so here I'm showing St. Paul in blue again and TLI in yellow. And you can see that yield on a per plant basis nearly doubled in the second year, along with reproductive tillers. And we had a serious drought at the Land Institute in the second year. So looking at the blue box plots provides the best estimate of what happens to yield between the first and second year. So this is in contrast to what we see in a sward environment where we have this substantial increase in yield. However, I noticed that a lot of my yield component traits decreased over time. So 1,000 grain weight decreased, yield per spike significantly decreased, and then all of my biomass traits also decreased. So flag leaves got smaller, stem diameters got narrower, spikelets per spike decreased, as well as florets per spikelet. So this tells me that those traits that are highlighted in green increase and those that are highlighted in red decrease. So this suggests that when we're looking at yield decline in wheatgrass, we should consider the decline in yield component traits. So we've improved our understanding of yield in space plants, which will better inform our ability to select for yield. And now I'm going to talk about one way that I sought to improve our ability to implement genomics-assisted breeding. And in order to utilize genomics-assisted breeding, you need to understand how your traits are controlled on a genetic level. And this can typically be done through biparental linkage mapping, in which you cross two divergent parents for a single trait of interest and associate genotype and phenotype in the segregating progeny. However, this approach has several disadvantages. For one, it's typically utilized for only one single trait for which the population was designed. Two, you have limited mapping resolution because you're only looking at the recombination events that occur within this single cross. So this approach called nested association mapping was developed to kind of overcome some of these challenges with biparental mapping. And in nested association mapping, you cross a series of parents by one common parent. So, and then develop multiple segregating progeny that all have one single parent, or that are nested within one single parent and have that parent in common. And this approach has demonstrated a lot of utility in other crops like wheat, barley, rice, maize, and so on. So we decided that because we have so many traits of interest in wheatgrass, and because the NAM can be utilized for all sorts of different traits in one single population, we decided to try it in intermediate wheatgrass. So from our University of Minnesota breeding program, we selected phenotypically divergent parents, some that were short and tall, some that had high and low threshability and high and low shattering. And we crossed them to a low shattering common parent and developed 10 of these segregating families. I harvested the seeds from these crosses, planted them out, propagated them, and then transplanted them in Minnesota and in Kansas in randomized complete block design. I harvested tissue and developed genotyping by sequencing libraries. And looking at the marker data that we collected from this population, we had just over 8,000 SNPs using the Genome Analysis Toolkit and the version 2 of the Intermediate Wheatgrass Reference Genome. And you can see here in the figure that these are each of the 21 chromosomes or linkage groups in intermediate wheatgrass. And each homologous group is colored in the same color. So this shows that we had excellent marker density across chromosomes and across groups. I'm sorry, a horizontal line indicates a marker, and the total count on the top is the total number of markers for that chromosome. So using that resource, we utilized, sorry, it's really early here in Washington. So we utilized genome-wide association mapping using those markers. And then I also worked with my colleague, Steve Larson, to develop a genetic consensus map with just over 3,000 markers using the genotyping data, and this allowed us to utilize linkage mapping as well. So looking at the marker PCA, colored dots indicate different families, and the gray dot in the center is the common parent, and the larger colored dots on the exterior are the donor parents. So those are the parents we selected that varied for those traits of interest, and their progeny are represented by the smaller dots kind of in the middle between the two parents. So this showed us that, gave us confidence that the population structure was what we expected with several biparental populations. So because this nested association mapping approach had not been done in intermediate wheatgrass, and nor had it been done in as obligate outcrossing species, we had to test it first. So we looked at flowering time, which was the first trait that was looked at in the maize nested association mapping population. And flowering time in wheatgrass is important to understand how it's controlled as we try to increase the, the diversity of the regions that we're growing Kernza, because optimal flowering plays a big role in determining yield, especially in other crops. So it's very important. So in my population, I looked at 2 different types of flowering time. The first I looked at was how far the spike emerged from the boot on a given day. And then I also looked at anthesis score. So I walked through the field when the plants were in anthesis and gave them a reading. And I'm going to show several figures like this. I'm just going to walk you through what this means. So on the y-axis here, I have emergence percent. So this is how far the spike was emerged from the boot. And on the x-axis, I have the 10 individual families and a box plot for each of them. The horizontal dotted gray line indicates the mean of the common parent, and the box plot distribution are the progeny within each family. And the median line in the box plot is actually the mean for each family. And the colored dots indicate the mean for the performance of each parent. And in the upper left-hand corner, I have both broad and narrow sense heritability estimates. And the higher those are, those indicate that the variation, a greater proportion of the variation we're seeing is due to genetics and less so due to environmental error. So some things to look at when I show a figure like this is, one, how high that heritability estimate is, which would give us power in mapping. And the second is how variable the families are, so how extensive the variation is within each of the families, and then also across the families. And then when I show all 4 environments, I want you to focus your eye on the fact that the trends with the performance of each family are relatively consistent across environments, which is also gives us promise and confidence in the phenotypic data. So you can see that the trend is a little different between St. Paul and the Land Institute, which makes sense because flowering time is very environment specific. So those are some things to focus on when I show a figure like this. So looking at the other method of assessing flowering time, the anthesis score that I rated, you can see also lots of variation across families and within families and consistent trends across environments. So looking at some of the results, this is how I will show them. So on the left, I have the results from the Genome-wide association mapping method shown in their physical positions. And then on the right-hand side, I have markers that were used in linkage mapping shown in their genetic positions, and markers that were used in common across both approaches are connected with a line. So when I show results on the left-hand side, those will be from genome-wide association mapping, and on the right, you'll see a bar or an interval that indicates the region of significance for linkage mapping. So looking at flowering time, I found a QTL for flowering time on chromosome 6 and for both emergence and anthesis, and this was detected in the St. Paul environment. And this same region was also detected in linkage mapping in all 4 environments, or sorry, in St. Paul, with both anthesis and emergence percent. And in flowering time, a major gene in barley, known as PPDH1, is also located on the equivalent chromosome in barley. And we were able to take that known sequence in barley and align it to the intermediate wheatgrass reference genome and figure out where it's located. And we were very close to this gene, indicating that this is likely a causative gene for influencing flowering time in wheatgrass. I also detected a QTL on chromosome 17 that was detected across multiple traits in multiple environments and across both approaches, which gave us a lot of confidence that, and this QTL was also, the same region was also detected in a previous study of maturity timing from my colleague Steve Larson et al. So in the end, we showed that looking at flowering time in the NAM, that it was under complex genetic control, and that many of the QTL we detected were environment specific. Furthermore, the QTL that we identified, one of them had been previously identified before, and another was a novel QTL that had not been identified before, but that aligned to a very likely causative ortholog from barley. So just despite the fact that this NAM approach had not been used in wheatgrass or in outcrossing, publicly outcrossing species before, we showed that it works in our species. So we decided to apply it to a couple more traits. So another trait of interest in wheatgrass is shattering. Shattering can result in yield loss, and it narrows the harvest window, and also can result in seedling emergence and contamination in a stand setting. So in the bottom right is a picture of intermediate wheatgrass field between the rows, and those little seedlings are a result of shattering. So I adapted a method from Lee for assessing shattering in my population. So I dropped spikes from 20 centimeters height for 3 different times, 3 different spikes per sample, and observed what fell off. And traditionally, this method counts the number of breaks that occur and the number of pieces that are left over. But I noticed, and if you look closely, you can see that what comes off the spike is actually kind of variable. So 2 things are happening. The first is the rachis is breaking. This trait is known as brittle rachis in other grass species. And the dispersal unit in this case is a large segment of the spike. And then also there's floret shattering, where the rachis, or the rachilla axis within the spikelet breaks, and the individual dispersal unit is a series of florets or a single floret. So looking at a correlation table of these 2 different methods of shattering, along with some various traits that I thought might be either morphologically related, perhaps something to do with physics. So like a longer spike maybe is more prone to breaking, and maybe heavier seeds result in more shattering. And then I also looked at maturity, trying to figure out if whether the variation I was seeing for shattering was really just a result of plants just being more mature than others. So again here, correlations that are significant are in bold, and correlations that are higher are in a darker green background. So some things to note here is first that brittle rachis shattering and floret shattering were not significantly correlated. So that suggests that they're independent of each other. Furthermore, there weren't any morphological traits that were super strongly associated with either type of shattering. And then finally, the influence of maturity was also very low and or insignificant, suggesting that the variation I saw was due to shattering and not just maturity. So looking at the variation in the floret shattering across families, you can see that we have a lot of variation within and across families, and that the performance of families as well as their parents was very consistent across the different environments. And looking at the mapping results, floret shattering was the most complex trait I looked at. It had the most QTL across the greatest number of different chromosomes. But I'm just going to highlight a couple examples here. So on chromosome 2, I found 2 QTL using genome-wide association mapping that aligned very closely to an ortholog from rice, SH5, and this region was also detected using linkage mapping. And then also detected a QTL on chromosome 11 for shattering, detected in multiple environments, and this same region was also detected in a previous mapping study, suggesting that there's something going on chromosome 11 for shattering in wheatgrass. And we're very confident that it's on the opposite end of the chromosome of a shattering ortholog from rice, SH4. So this is likely a unique method of shattering in wheatgrass. Looking at the brittle rachis type of shattering, again, lots of variation. You can see by the horizontal dotted line that the common parent was actually quite susceptible to shattering, to brittle rachis shattering. And the 2 different regions that we detected for brittle rachis shattering were on chromosomes 7 and 8 across multiple environments. And we were very close in proximity to the orthologs of the brittle rachis genes from barley, BTR1 and BTR2. So I've only got a couple minutes left, so I'll probably just proceed and maybe take questions via email. Got a couple more minutes here. So I want to mention that we also looked at threshability. So I had the opportunity to travel up to Roseau, Minnesota and look at what was coming off the combine and talk to a grower about his seed cleaning operation. It was very complicated, and he's like, it would be great if wheatgrass shattered better, or sorry, threshed better, cleaner. So I looked at harvested spikes from wheatgrass, threshed them out really in a Wintersteiger LD350 laboratory thresher, and rated the percent clean seed using known standards in Petri plates. So this is an example of what I saw: 20% threshability, and in some cases 100% threshability. And overall, my population had a high threshability rating. So either it was the thresher I was using, or my population just happened to be very free threshing. And the threshing ability in wheatgrass has often been described as a similar mechanism as in barley, or the naked locus or nude on chromosome 7, which is equivalent to intermediate wheatgrass chromosomes 19 through 21. However, I did not detect any QTL on chromosomes 19 through 21 for any of the environments I looked at, nor in any of the families that I looked at. And also, previous study looking at shattering did not detect QTL there either in wheatgrass. So I'm losing confidence that at least now this type of shattering is segregating it was segregating in my population, and maybe has been purged in the breeding program. But I did detect a QTL on chromosome 5 that was detected across both approaches in multiple environments. And in wheat, the type of shattering is very different than it is in barley, and it's controlled by the soft gloom and tenacious gloom genes, which are homologous to the chromosome that I detected shattering on. So my feeling is now that maybe this type of shattering is segregating in our populations and may be the target of selection. So I showed that we can utilize some of the findings from my work to further implement our genomics assisted breeding efforts. And now we have a greater understanding of yield, at least in a space plant setting, which can inform how we select for yield. And I also just want to put in a little plug for all my data, which is on the intermediate wheatgrass database. Thanks to Jared Crain for his help on that. It's available for anyone to use. I've got sequencing data, genetic map, and input files for map QTL, lots of stuff that can be looked at here. So please reach out if you're interested in collaborating. So thank you. Sorry, I'm a little over.
You are perfect, Kayla. To the second. Can you click to end your show?
Yes.
Great, fantastic. Thank you for that excellent presentation, Kayla. We are out of time, but I think we'll try to get speakers' email addresses out to participants if everyone approves, so we can perhaps continue the questions by email if necessary. We're continuing our breeding session. Sorry to everyone to dive into genetics and breeding right off the beginning here. It goes back to the days when this was in person, and we tried to get the genetic stuff when people were fresh in the morning and able to slog through some of this material. It's exciting to us geneticists and breeders, at least. So next up is Steve Larson, who has worked for 22 years as a research geneticist at the USDA ARS Forage and Range Research Laboratory in Utah State University in Logan. He's worked on the genetics and breeding of intermediate wheatgrass and other perennial grasses. Steve's been a great collaborator over the last several years since we started working on intermediate wheatgrass together. He's going to talk to us about preliminary results from his breeding project that he is starting now in Utah. So Steve, you can go ahead and share your screen if you'd like and get started.
Okay.
You are sharing. And it looks great, Steve.
Okay.
Go ahead.
Good morning, everyone. I want to start by saying that I'm grateful to all the people listed here that made this project possible. Doug Cattani, Jim Anderson, and Lee provided plant materials to get this started. Jesse Poland has been instrumental in developing the the genome sequence that we're using, the breeding database that Kayla just mentioned, and laboratory methods that I'm using. Jared Crain has been responsible for developing pipelines for data analysis that I'm using, and extremely helpful and patient in showing me how to use these pipelines. The USDA Cycle 0 base population is comprised of 77 plants from each of 3 sources, institutional sources, and 29 families as listed here. I will describe and compare sources and families, but it should be understood that these samples do not represent the entirety of these respective programs. And I would also point out that I'm only testing these materials in northern Utah, which is a much different environment from Manitoba, Minnesota, or Kansas. This figure shows the first 2 principal components from a PCA analysis of DNA genotypes from 2,233 plants. It shows good separation of all 3 institutional sources, especially the Manitoba materials seen here on the right side of principal component 1. This figure shows a PCA analysis of DNA genotypes from 770 plants from Manitoba, showing separation of groups within and among the 10 lines. I assume that these lines were identified according to 10 different female parents, but there are more than 10 groups here, which may reflect the complexity of having at least several well-defined pollen parents. I will come back to this idea in a few minutes. This figure shows a PCA analysis of DNA genotypes from 385 plants from Minnesota, showing pretty good separation of the 3 lines, namely 1501, 1505, and 1504, and a group of 2 lines here in the lower right, 1503 and 1502, that look similar. I believe the 1504 line seen here in dark blue was recently released as Clearwater variety, the first Kernza variety. This pattern of grouping makes sense if the populations were isolated and randomly mated. This figure shows a PCA analysis of DNA genotypes from 1,078 plants from the Land Institute, showing considerable diversity within lines and some differentiation among the lines. It's similar to what we saw with the Manitoba material. This table summarizes paternity analysis of the same 1,078 plants, which came from 14 seed parents and 89 possible pollen parents from a crossing block at the Land Institute. The 14 plants, the first 14 plants highlighted here in yellow were used 77 times each as the seed parents of these 1,078 plants, and at least 73 of these 89 plants were used at least once as the pollen parent, and there were two of these 89 parents that were used over 160 times as pollen parents. So the plants are not utilized as pollen parents equally or even randomly. And I think this, if the number of plants is small, say 10, and only 2 or 3 were utilized as pollen parents, then I think that might explain the patterns we saw for the 10 lines from Manitoba, which did not really show a perfect grouping of materials by the half-sib family name, or the presumably the maternal parent name. We have finished measuring 4 traits on the training population, but we only got about 1,000 measurements of seed yield and seed mass before the mice destroyed my harvest bags. However, we still have a nearly complete set of inflorescence samples that will be used to measure additional spike and seed traits that Kayla mentioned earlier. We pushed forward quickly on trying to get as much seed from each plant as we could so that we could plant this experiment here. We just finished planting 2 reps of 900 half-sib progeny lines for plot yield evaluations. If you look carefully at this photo, and we just did that planting a couple weeks ago, and if you look carefully, you can see seedling coming up in the rows, and you can even discern separation of the plots over here in some of these rows. All pairwise trait correlations were significant, but not particularly strong. The strongest correlations between just these four traits were between plant height and seed yield per plant. There was a weak negative correlation between head emergence and seed size, meaning that early plants had smaller seeds, but that was obviously not a very strong effect. We saw significant heading differences among the 3 sources and 29 lines listed here. Plants from Manitoba were slightly earlier, but the range of values, well, they were significantly earlier, but the range of values was actually quite narrow, from 58.4 to 57, really essentially 58. So only a half a point difference there, half a unit difference. With nearly 38,000 markers, we detected two or possibly 3 chromosome loci associated with heading. I could not immediately identify candidate genes for these effects the way that Kayla was able to, but it will be interesting to compare these results with other Kernza genetic studies, including Kayla's and Jared's that we'll see later today. The heading predictions based on ridge regression ridge regression blobs were highly correlated with observed values. So the predicted values on the y-axis, observed values on the x-axis, and this was using a random 5-fold cross-validation. So there's a very, very good correlation between predictions and observed values, at least for head emergence. This figure shows correlations of predicted and observed heading values only for Manitoba plants, using only Minnesota plants or plants from the Land Institute as training populations. So here's the correlation of predicted and observed values using Minnesota lines as a training population, or using the Land Institute plants as training population to predict values of the Manitoba population. These correlations are significant, but the regression lines from the 2 different training populations are not the same, and there's considerable bias in these predictions, especially with higher values. There was also significant plant height differences among the 3 sources and 29 lines. Reduced height is probably desirable for a small grain crop, and some of the shortest plants were from Manitoba and the Minnesota 1504 line, which was released as Clearwater. So those are some of the shortest lines. Plant height predictions were highly correlated with observed values using the random 5-fold cross validations for plant height. This figure shows, again, correlations of predicted and observed plant height values for the Minnesota plants, using only plants from Manitoba as the training population, or only plants from the Land Institute as training populations. Again, the regression lines the slope of the lines are a little bit different, and there's some bias again, especially at higher values in those predictions using the two different training populations. There were significant seed yield differences among the three sources and 29 lines, with two of the three top lines from Manitoba for seed yield, a high yielding plant line with reduced plant height, like this Manitoba 152 line, could be useful. We detected two chromosome loci, chromosome 6 and chromosome 4, associated with seed yield. We also detected a lot of seed yield QTLs, or seed yield components, in the QTL analysis of the M26, M35 population, which I previously have reported on. Seed yield predictions were correlated with observed values using random fivefold cross validations, and it seems, and this seems consistent through a wide range of values. This figure shows correlations of predicted and observed seed yield values for plants from the Land Institute, using only plants from Manitoba or only plants from Minnesota as training populations. Both training populations give similar regression lines in terms of slope, but there's about 5 grams of difference in or bias in these predictions using the two different training populations. Finally, there were significant differences for seed mass among the three institutional sources and 29 lines, with most of the top lines coming from Kansas in this case. The best line had an average among the 77 plants from that line of 10.2 milligrams per seed. Seed mass predictions were also correlated with observed values using random fivefold cross validations. So really all of the random fivefold cross validations were good for all 4 traits. This figure shows pairwise cross predictions from one training population to one prediction population. So you can see the slopes of these lines for seed mass are different depending on which training population and which predicted population are used in each pairwise comparison. I think all these correlations are significant and potentially useful, but if you put all these predicted and observed values in one data set, the overall correlation was pretty close to 0. So yeah, and that's presumably because of the differences in the slopes of these lines, as well as bias between the different predictions. This upper table summarizes all cross predictions using one training population, Manitoba, Minnesota, or Land Institute, or 2 training populations on these 2 lines, or these 3 rows, against each each of the 3 possible prediction populations and 4 traits. And one of the patterns that seems to be, that emerges here is that the Manitoba population was often the most difficult to predict, especially using the Minnesota training population. But if you use 2 training populations, in most cases, the correlation of predict and observe values are pretty good. But again, for seed mass especially, the materials from Manitoba were the most difficult to predict, regardless of what training population is used. And that the Manitoba population is different in that it's earlier, a little bit earlier. They tended to have lower stature and higher seed yield, but lower seed size compared to the Lannister population, which had high seed mass and high plant height. So I will
finish, Steve?
conclude by thanking my all of my collaborators, coworkers, institutional supporters listed here. In addition to those that I mentioned earlier, Lynn Johnson has done most of the DNA work here at the Forage and Range Lab, the greenhouse work, and many of the trait measurements. You saw Trapper, Chloe, and Josh doing field work in some of the photos. In addition to all the help that I got from Jared to analyze the data, I also get a lot of critical support from Matt Robbins, who is an expert in bioinformatics and high performance computing systems here at the Forage and Range Lab. Finally, I thank everyone who joined this meeting this morning.
Great, thank you very much, Steve. I don't see any questions yet. We have a couple, or a half a minute maybe. I have got one question for you, Steve. On the data coming from these different locations, you say there's kind of a bias because there's different values, I assume, on average at those different locations. Is it possible to standardize those by taking out a population average before generating the predictions, perhaps?
Well, yeah, if you know what they are, but if you don't, if you had no data from one of the predicted populations, then I'm not sure how you would do that. If you have all of the data, if you use all of the data, yes, I believe you can make good predictions.
So yeah, one analysis to throw it all in at one time, basically.
Right.
Splitting them apart.
Right. If you use real data from all 3 source populations as in your training, then yes, the predictions are good.
Right, great. Thanks, Steve. You can unshare your screen. And I'll ask Jared to start sharing his screen here. Hey, Jared, you can go ahead and start sharing. Great. I'd like to introduce Jared Crain. He's a postdoctoral researcher at Kansas State University and collaborates with me directly here, working on really processing and analyzing a genomic selection and data analysis and storage and manipulation for my breeding program primarily. It's been a great benefit to me and come up with some excellent analyses. Looking forward to what Jared's going to tell us today about looking at GWAS, or Genomic-wide association studies, on some of the materials we've been looking at in the breeding program over time here. So go ahead, Jared.
Okay, thank you, Lee, for that introduction. And I think I've got my slides up. Tell me if I don't have them.
It looks good, Jared.
Today, I'd like to go over what I'm calling preliminary results. I feel pretty confident in them, but as they continue to get further refined, the exact values you see here will probably change. But just to start with, I wanted to give I know we've got a broad audience, agronomists, food scientists, not just the geneticists and breeders, so why we would be interested in a genome-wide association analysis and what we're trying to do with that. And the real purpose is we're trying to figure out what areas of that genome are controlling our traits of interest, whether that's spike yield, free threshing, chattering, just like Kayla was talking about. And then from there, we can use those results to inform marker-assisted selection to preferentially maintain or eliminate alleles from the breeding population, and to kind of from a theoretical sense to better understand what the genetics and the biology of the intermediate wheatgrass is. And we'll see a lot of these plots. This is just a Manhattan plot. It's a stock photo off of Wikipedia. I chose this one because it shows the ideal plot when you think of GWAS. And for everybody, what we're seeing on the x-axis, numbered 1 through 22, this is the chromosomes. And then each one of those dots in the plot is one SNP marker where it's been tested to the phenotype. Is this SNP marker associated with the phenotype of interest? And then there's dashed lines signifying the level of significance of that association. And for this particular trait, over on chromosome 19, you see a really high peak, suggesting that there's something on chromosome 19, particularly around these markers, that is controlling that trait of interest. There's other peaks as well on chromosome 12 and 6. But I just wanted to give an overview so that as we look at these Manhattan plots, everyone can get a feel for what we're looking at. So there are several challenges of doing a genome-wide association analysis in the intermediate wheatgrass. One of the first things is this plant's very heterozygous and outcrossing. So that means there's lots of opportunity for recombination and that we have a low linkage disequilibrium. And this is a figure from a paper that Xiaofei Zhang published in 2016, looking at the extent of LD in some of the Minnesota breeding population. And with this figure, genetic distance is on the x-axis, so increasing genetic distance as we go toward the right, and the degree of linkage disequilibrium is on the y-axis with 0.75, these higher numbers, meaning that the SNP markers are linked together and they're not randomly placed throughout the genome, that they are in non-random association. Prabin has also looked at and made a figure very similar to this with the later UMN data. And then this is a figure that I've worked up in the TLI breeding population. It is on the x-axis, this is distance in base pairs. But again, on the y-axis is that level of linkage disequilibrium. And we can see that at a very, for the most part, we have very low LD, that to find markers in high LD, they have to be very close together. So this suggests that we need lots of markers to cover that genome. So besides needing lots of markers to identify traits, we also need genotype by environment interactions, or another challenge with these GWAS. Some of the first results I looked at, and I'll just show 2 traits. This is spike yield. Again, our Manhattan plot. We have some SNPs on chromosome 8 that are above our significance threshold in 2016, and I've highlighted those. In 2017, this trait actually showed similar expression. We still had SNPs on chromosome 8 that were were significantly associated. So this is what we'd like to see. This is probably some gene that is actually controlling that trait. But a lot of traits don't actually show this same type of pattern. For example, percent free threshing in 2016, across the genome, we had scattered markers that were statistically associated with those traits. And then in 2017, we had the same data, but you can see right away, and I've highlighted a lot of them, that the markers don't have the same values. That, for example, the green box on chromosome 1 shows a non-significant trait in 2016, yet there's a significant trait association in 2017. And just as an example, the blue box in 2016, several SNPs across chromosome 4 through 11, none of those end up being significant in 2017. So this is really challenging to get this data and look at it. Am I really actually seeing something, or is it just an artifact within the data? So what we'd like to find is something that's biologically relevant, that's not just an artifact from one particular environment, year, cycle of information. So what we've been working on is using multiple years of data. The phenotypic data ranges from 2016 to 2019 over 3 cycles of 3 different generations, using about 22,000 SNP markers. What we've done is analyzed all the years of data, so spike yield year 1 and year 2 for cycles 6 and 7, and spike yield of year 1, cycle 8 was just from 2019, so we just have 1 year of that data. And this is the breeding program data that we're using, starting with TLI cycle 6. There's about 2,200 observations. This data was collected in 2016 and 17. And then we started genomic selection, which we'll see more tomorrow with that, in 2017 with cycle 7. And we had our first year of data observed in 2018 and 2019. And last year, cycle 8 was coming online with about 1,100 plants. So in total, we're getting up to about 4,000 phenotypic observations. So this hopefully is giving us some power to detect those genomic regions of interest. So using that information, I'll go over the current results that we have. This is brittle rachis, just like Kayla was talking about. Across the 21 intermediate wheatgrass chromosomes, our best significant markers came up on chromosome 7 and 8, which is homologous group 3, and group 3 of the barley genome is the brittle rachis. So this gives some confidence that what we're actually, that these models are actually giving us real biological information, that we're not just seeing an artifact or garbage out of these GWAS models. And then another trait, a seed width. This one just had a very nice, on chromosome 10, lots of markers all going up into a peak together. We would probably like to see, compared to that ideal peak that I showed starting with, we don't see that. That could be a very big function of marker density, where we're only using 22,000 markers, and based on research from Xiaofei and Prabin, LD decays almost by half at 1 megabase or less. So we need lots of markers to be able to detect those associations. And then the last kind of agronomic trait that I'll show before we get into yield components is plant height. This one we didn't find at a significance level any markers that were significant. However, our highest scoring marker is on chromosome 11. And Steve Larson has data that within this homologous group 4 is where the RHT, the reduced height green revolution gene is. So our best hit does follow along with something that we would biologically conceive as quite possible. So again, more evidence that our models are actually giving us good data. So the last thing that I really want to discuss is the yield components, and to kind of set the background up for this, within the TLI breeding program, the cycle 7 population as a whole yielded about 0.51 grams per spike in yield. Those cycle 7 parents that we selected, or cycle 7 individuals that we selected to be parents of the cycle 8 population, ended up yielding about 0.62. And this was all done by genomic selection. And ideally, when we look at this, we think this is great. This is the direction we want to go. We want to be driving that yield higher. And by all indications, we are. However, when we start decomposing that spike yield, there's at least some indication that we might want to study this closer, particularly looking at percent seed set, as Kayla mentioned, floret site utilization. In cycle 7 individuals, as a whole, we had 35% seed set. When we look at those selected parents, we were only at 33% seed set. And that trend kind of follows throughout. If we look at number of florets per spike in our parents, the number of florets per spike went up compared to non-selected. So this plant is making, it is yielding higher seed, but it's doing so at the expense of making a lot of more components that it's not actually setting seed on. So ideally, we would actually like to see this, our percent seed set increasing, and that plant being more efficient in the floret structures and the reproductive structures that it makes. So using GWAS is one way to start to identify what's going on, and is making sure that we can breed for higher efficiency and higher yielding together. So the next few Manhattan plots just look at spike yield and those components of yield. We did see a significant marker trait association for spike yield on chromosome 13 and 18. At this point, we haven't went through completely into the literature to see if we can identify most likely candidate genes with these. And then when we start looking at yield components, number of florets per spike, several strong peaks associated with chromosome 6 and 10. And then this is number of florets per spikelets. For each individual spikelet on that inflorescence, we do see again strong peaks on chromosome 6 and 10 and 9. So we are identifying several areas within the genome that are at least look like they are associated with these traits that we're wanting to measure and identify. Seeds per spike, we had one significant marker on chromosome 13. And then there are percent seed set. Again, this is the one that decreased to 33% from 35%. We didn't find any significantly related markers, at least using our correction thresholds. Going forward, we can keep examining this. And spikelets per spike, this completes all of the individuals that were shown in the table. So going forward, future direction, we found several significant markers near known genes. Looking at some of those, we also know that we have several significant markers for different yield components that we'll go through and try to identify literature search candidate genes and other known genes that are out there that would be affecting those traits that we're measuring. And looking long-term into the future, hopefully new genotyping methods, instead of using a 20,000 markers, we'll be able to use 40, 50, 60, even over 100,000 markers to identify some of these. It's going to give us a higher resolution and precision to identify these genome regions of interest and to be able to use them in the breeding program. And right now there's, at least within perennial rye, there is literature out there that they have increased grain yield while also increasing those yield components, floret site utilization. So at least based on literature, it doesn't look like it's one or the other, that we either have high yield and inefficient plants. It looks like we can do both as we start to understand these traits better and make selections on them. And with that, I would like to acknowledge this work's been the collaborative effort of a large group of people and funding through Kansas State, the Land Institute, and the Malone Family Land and Preservation Foundation. So with that, I'll turn it back over if we have time for questions, or Lee, I'll let you moderate that.
Yeah, Jared, thank you very much. Great presentation. And we do have a little time. If someone wants to type in a question, you can go to the question box at the bottom and type in a question, and we can deliver that to Jared. Looking if anyone's sending any down there. Not seeing any popping up at the moment. I could ask you, oh, here we go. Somehow popped up. From Nathan Tintle, Jared, are you also thinking about a pathway method for GWAS?
By pathway, is that similar to what Kayla was talking about with the?
I'm not sure.
I'm gonna answer. I'm gonna go that way.
Nathan says gene ontology, IPA, etc. I think is what he has in mind.
We are looking for candidate genes, and those locations are significant markers. We will see what genes are around there, if any of those make sense for the traits that we're observing.
Another question from Matt Baker. Aren't there environmental effects that are confounded in contrasting years in this approach?
So all of the data, and I glossed very quickly over the model, we do have effects for year and the cycle and harvest number for those plants, that hopefully we are accounting for that, that what we're seeing is an actual prediction value of the genetic potential. But it is definitely a very good question, and something that I think we're accounting for it, but it's a very challenging within the intermediate wheatgrass because it is unreplicated as well. So.
All right, thank you, Jared. Our time is up.
Yeah, thank you.
We will move on to our next section in our schedule this morning. First section was called genetics. The second is breeding. Slight distinction there, perhaps. A lot of overlap, however. I see Prabin is here. You can go ahead and start sharing your screen if you want, Prabin.
Yeah, sounds good.
So I'll introduce him in a moment. Prabin Bajgain is a research associate at the University of Minnesota, where he manages the intermediate wheatgrass or Kernza breeding and domestication program. He's been working on crop genetics improvement using breeding and bioinformatics and applied genomics since 2009. He collaborates with multiple teams with diverse expertise within and outside the University of Minnesota to improve intermediate wheatgrass genetics, agronomics, in-use, marketing, and he would be happy to talk to you by email also if you're interested in connecting with him more after this talk. Go ahead. I see your presentation there, Prabin, so take it away.
All right. Hope everyone can see me and hear me fine. Great. Thanks, Lee, for the introduction. In my short talk today, I'll be sharing with you some of the results we have from a recently completed project where we looked at how using GBI or genotype by environment interaction effect, how does that affect genomic prediction in wheatgrass? You heard some of that from Steve, some from Jared, and hopefully I'll be talking about the GBI in a bit more depth. And this will be in relation with only the University of Minnesota population. There we go. So first of all, some background on genomic selection to those who are not aware of what it is. It's basically an alternate or a supplemental method of selection where you essentially use genome-wide markers to make predictions and selection decisions on a large population. So you start with what we call a training population, you can see on the top left, a smaller subset of your actual breeding population that's phenotyped for traits of interest in multiple locations, hopefully. And then you also have genome-wide markers, several thousands of them. And then you estimate the marker effect shown in beta in red there. So you heard about a GWAS or association mapping in previous presentations. The model does essentially the same thing, but instead of pointing out your significant loci, the model just assigns a weight or an effect to these markers. Then you take those markers and apply onto a larger breeding population that has not seen the field yet, just genotyped with the same markers as before. And then you obtain Y, or the phenotypic trait value. So based on these predicted, genome predicted values, you make your selections, and then you can either use those selected genotypes to make crosses or evaluate them in the field, depending on your breeding design. But the whole idea is to save time and resources by evaluating a larger population just based on marker that is trained using the phenotype data on a smaller subset. So with that in mind, let's look at how our trials are set up. We have two locations in Minnesota, one in St. Paul where we're located right now, and one in Crookston that's northwest, about 80 miles south of the Canadian border. This particular population that I'll be presenting today had about 550 plants from 70 families, which is about what we have for other populations too. We typically establish our populations mid to late September, and then we fertilize with about 50 pounds per acre of nitrogen end of April or beginning May, depending on the weather. And we measure a lot of traits. Most of those you've already heard, and you can see on the slide, I've mentioned them here quickly. The figure and the picture on the right, middle right, is our St. Paul location. Bottom is Crookston. They're actually the same genotypes cloned, but the pictures are taken in different times, so you can obviously see that there are different stages of plant growth. These two sites vary quite a bit, actually, despite being in the same state. The soil type is different. Crookston has fine sandy loam, whereas St. Paul has silt loam. The amount of precipitation is also different, as shown by the heat map on the right. St. Paul definitely gets a lot more rainfall and snow overall compared to Crookston. Then we look at day length in those wavy pictures on the left. Crookston typically has longer days, shorter nights compared to St. Paul. The temperature is also different, and that is indicated. I don't have a great picture for this, but you can tell that Crookston in general is colder than St. Paul. So quite a lot of variation, and you can expect the plants to do differently in these two locations. And to prove that point, here's a chart of correlation of several traits on the left. There's a lot of numbers, but I want you to see or look at those red square boxes. Those are correlation values for 1,000 kernel weight between these two locations in 3 years. The values are fairly good, 0.51, 0.54, and 0.73. So not too bad compared to some of the other ones where we have negative correlations or close to zero. However, despite having good correlation, there's very strong GBI, as shown by the table on the right. I have sorted the best 20, or the genotypes with the largest seeds in St. Paul 2017, but then if you look at the rankings of those same individuals in St. Paul 2018, a year later, and Crookston 2018, you can see the ranking differences are quite crazy. You will not find top 20 or even a similar order there jumping all around the place. and a very strong indication that the environment is affecting how these traits are expressed in these locations. To prove that point further, I've talked with some of you about this phenomenon. These single plant pictures are from Crookston, not great pictures, but we can kind of make out that in both these single plants, about half, if not more, of the tillers are completely dry, very ready to be harvested. However, there's also about half of the tillers that are very green. This was taken, I think, August 12th last year. And if you do not know, that's prime time for harvesting. So these plants are still trying to pollinate and make more seed. And it's very interesting because we do not see that in St. Paul. It's something that happens just in Crookston. I've seen it in the past 2 years. I'm not sure why this happens, but probably because of the photoperiod, the temperature that has something to do with it. So this is something we're dealing with. And then once I saw this happening between the two locations, I thought, well, why not take into effect this G/E interaction and try to do something with it? But before that, why do we care about G/E? Well, we care because we cannot ignore it. You can talk to any breeder, any crop, even crops that are planted just a few miles apart, we'd have different trait values because there's environmental differences. You heard from the speakers before about complex quantitative traits, which wheatgrass tends to have quite a lot of those. These traits are actually more affected than, let's say, traits affected by or controlled by major genes. And if you are breeding for variety development that farmers will be growing in different regions of the state or even the country, you have to consider these G/E effects so that you develop varieties that perform relatively well in different diverse locations, especially with this unpredictable climate that's happening around us. So quick look at the prediction models I used. These are not my models. These are developed, I think, initially proposed by the CIMMYT scientists and later incorporated into our packages by other people. I used 3 different types. The first one was MM, which looked at the main genetic effect across all environments. Second one was MDS, excuse me, which takes into account the main genetic effect for all environments and a single G/E for all environments as well. And then we have MDE, which takes into account the main genetic effect for all environments and a single G/E for each environment where the plants were phenotyped. So in essence, 3 different tiers or levels of model design. The values in the bottom are heritability values. We'll be revisiting them later as I show you the results, but I want you to sort of kind of get an idea of these broad sense heritability estimates that vary quite a bit, all the way up to about 0.72 in 1,000 kernel weight. I think Steve had as high as 0.8. So that's a good trend that we're seeing across our program too. There's numerous studies in other crops, other programs that show that when you use G/E, it helps boost predictions. And these are multiple crops in different programs. The numbers in green are the highest bump in prediction accuracies when they included G/E effect into their models for any given trait. I'm just highlighting the highest increase they saw in these studies. So that was exciting, and that's why I implemented those models and looked at our traits. So if you look at the bar plots, the ones in red border, the dotted red border to the right of each block are the highest values obtained when G/E was not considered. For some traits right off the bat, even without G/E, you can tell that the values are quite high. Whereas for others, without G/E, they're low, but with G/E, they're high. I'm to highlight some. For spike weight, I saw an improvement by 15%, which had heritability of 0.69. For spike length, about 20% boost despite having low heritability, so that was very encouraging. For freeze threshing, the improvement was 23%, and that also had low heritability. And then about 18% for grain yield, which had relatively high. And then for others that had different heritabilities, the predictions didn't really improve a whole lot. But the takeaway from this is that when you use G/E, for heritability, for traits with heritability estimates low or high, you can improve your predictions. So I used the models from the previous result and then looked at what happens if we apply those models to predict these traits in different environments. And you saw, I think, some of those from Steve already, where he looked at predictions based on Minnesota, Manitoba, or the Land Institute. So here's 3 that I'm highlighting right now where, remember that, there's heritability values at the bottom. So when you have a trait with high heritability estimate, you can make, in general, sound predictions using only 1-year data. And particular, if you use a first-year trial, you can predict the second-year trials relatively well. But then there's traits that just act crazy, no environment is able to predict other environments as well. And then there's other traits not highlighted here, but if you look on the very left of the figure for spike weight, it has high heritability estimate, but the predictions are overall fairly low. So it does vary by trait, but in general, if you have high heritability estimates, you can use certain environment to predict other environments. So the takeaway from this result, I have 2. One is that if for some reason a trial goes bad or you have to abandon it from natural or a man-made disaster, then we can still predict how the plants will perform in that environment using other environments, especially for the traits with high heritability estimates. And the second is that as we know that wheatgrass breeding is now expanding to multiple states and countries, we could probably make relatively sound predictions on some of these traits using trait data from other programs. And again, going back to Steve's presentation, I think that was an excellent display of what happens when you combine the data sets of 2 programs to get relatively high values and make breeding selections in case you have to abandon the trial or don't have as sound data. So going back to the question, can G/E interaction effect improve prediction? Yes, it is trait dependent. The improvement varies, but overall, I would say it very much improves prediction. Now, before I end, I'd like to also discuss the use of dominance effects in predictions. You heard from Jared about high heterozygosity and outcrossing. My thought was wheatgrass being an outcrossing, highly heterozygous population, is some of those heterozygous loci adding anything towards dominance, and is that dominance doing anything in genomic prediction? So this is the composition of our cycle 3 population, where you can see almost 40% of the loci were heterozygous. And I was also inspired by publications that showed that using non-additive, mainly dominance and epistatic effects, improve predictions. Unfortunately, that was not the case for us. Additive dominance made no difference. I think with dominance, I saw improvement of about only 3%. I forgot the trait, but in general, there was no great boost. So for at least Minnesota populations, I don't think we need to focus on dominance, but maybe it'll be different for other programs in other countries and states and other traits for them. With that, I'd like to end by acknowledging there's a lot of people who, whose work has gone behind making this project a success, and mainly the funding partners from the Forever Green Initiative at the University of Minnesota, the General Mills Foundation, and Minnesota Department of Ag. I don't think I have time to take any questions, but I can take 1 or 2, or maybe keep it for the end.
Yes, Prabin, it looks like you do have a few minutes left here. So definitely encourage people to go to the bottom of your screen, click on questions, and submit a question for us. See one coming from Anna Westerberg. Did you use single replication of the genotypes at each site? If so, would several replications within each field affect your results and also improved genomic prediction?
That's a great question. Yes, like Jared said, we also do single reps, mainly because it's a challenge in cloning and managing these perennial plants in multiple reps. We do have checks that I use to sort of take out the error variance from the environments. I do believe having reps would boost these predictions, probably by 3 percentage points. But that would be something to test if we had, let's say, a smaller population of only a few hundred and not larger population.
So at these two locations, though, you had replicated genotypes across the two locations?
Yes, those were the exact same genotypes, but within each environment was only one rep.
Any other questions? Waiting to see if any come in. All right, seeing none.
All right, well, thanks
Okay, move
everyone. I can be reached by email if you have any questions about the methodology.
Thank you very much for your presentation. That was excellent, Prabin. All right, we can move along to our next presentation. Also from the University of Minnesota, we have Jim Anderson coming up. And Jim Anderson is a professor in the Department of Agronomy and Plant Genetics at the University of Minnesota. He leads crop improvement programs focusing on spring wheat and intermediate wheatgrass, or Kernza. And his research on each species includes cultivar development, investigation of breeding and genetic principles related to crop improvement, identifying genes underlying economically important traits, implementing marker-assisted and genomic selection for improving crops. All right, with that, Jim, I see your presentation is titled Intermediate Wheatgrass Breeding and Domestication at the University of Minnesota. I know you have a brief time here, so I'll let you dive right into it.
All right, thank you, Lee. So what I'm going to do is just kind of give an overview of the breeding program. So Prabin, who you just heard from, is really operating this breeding program, but we've split up. So he talked about the genomic selection work, and I'm talking about the breeding part and cultivar release. I have to figure out how to advance here. Okay, so I think many of you have seen a slide similar to this that we've produced in the past, showing kind of a brief history of intermediate wheatgrass breeding. So you all know it started out as a forage species. USDA, Rodale Institute started working with it, and then the Land Institute has had a breeding program since 2002, and they've completed, I believe, 8 selection cycles now. University of Minnesota started breeding intermediate wheatgrass in 2011, and those materials came from the Land Institute as 66 mother plants, 2,560 seedlings. They were planted out in the field, and we are currently in our 4th selection cycle. And the main feature of our breeding program is we did release a variety, MN Clearwater, in 2019. And the source plants of that variety actually were selected from the 1st selection cycle. So it took several years just to do the seed increase. So just so you have an idea of where Clearwater is coming from, that was not a lot of selection at Minnesota, just one cycle of selection that resulted in those plants, source plants. So these are the priority traits that we're breeding for: grain yield, sustained grain yield, because what we've learned over the short time that we've grown this crop in Minnesota is that grain yield declines precipitously in year 3 of production, and we actually see some decline in year 2 as well. And we're exploring agronomic solutions, but also mindful of how breeding can contribute to sustained yields, perhaps selecting for less competitive plants that will maintain that yield over time. Other traits: seed size, shattering resistance, free threshing. These are all traits that would help in the end use characteristics as well, reduce the cost of growing and producing the crop. In Minnesota, the plants can get fairly tall because we are a fairly high rainfall environment, and when we get the summer rains and thunderstorms, we get wind associated with it, we can have lodging problems. We're also mindful of potential diseases. We've screened for Fusarium head blight and sort of decided that it wouldn't be too much of an issue. We would call the germplasm moderately resistant overall, but there were some issues with high vomitoxin in the grain in some of last year's production field. So we're monitoring that as well. And we also see some ergot in the crop. So this is the kind of the 30,000 foot view of the breeding program. Prabin talked about the genomic selection. So the figure that he showed is really encapsulated here on the left-hand side. We have a training population that we phenotype in Crookston and St. Paul. That's a subset of a larger breeding population, so around 600 plants. That data goes into a genomic selection model that's used to predict the performance of 3,000 plants. And then from there, we choose parents to be used for the next cycle of breeding. And so every year we are developing new variety candidates, and we are producing what we call synthetic varieties. So these are a few selected individuals, usually between about 7 and 12, that are intercrossed, and we increase seed for a few generations, do yield trials on that seed, and maybe perhaps another seed increase generation, and that results in a variety release. So quite a bit involved here, and I'll talk a little bit about more about this in a minute. So what we've learned, what we've done after 4 breeding cycles, the amount of seed shattering has been reduced. Threshability is on the increase. So this is the seed threshing freely from the hulls. I think in later seminars you'll hear that Lee's got some good advancements in threshability also. Seed size is on the rise. On the left here, these are seeds of rush. The next two are a couple of samples of MN Clearwater harvested from different locations. But I also want to show these next columns. We do have other large seeded materials within our breeding program. I noticed the MN 1501, which is our largest seeded variety candidate. We did not release it, but it also showed very large seed in Steve Larson's trial that he presented earlier. So we've got a ways to go. Here's a hard red spring wheat laying MN, considerably wider, about 2 to 3 times the seed size of our typical wheatgrass at this particular time. So MN Clearwater was released in 2019. It's producing an average of 696 kilograms per hectare of grain. And this is over 1 and 2 years, first and second year seed production. It's available to growers in a limited public release. You'll be hearing more about that from Colin Curitin. He's going to be giving a seminar June 5th, 9:30 a.m. Colin is a supply chain development specialist with the Forever Green initiative. We have prepared a plant registration article. It's available in the Journal of Plant Registrations. I believe it's called the Early View, and the journal is transitioning to a new publisher, so I actually couldn't get the full version of the article, and maybe that's because it's in Early View. But you can be looking for, you can at least see the abstract, I know, in that journal. Some data on this variety. So it was tested along with our other synthetic variety candidates. So these are the other MN numbers and a couple of TLI selection cycle materials, and Rush, which is the old forage variety. So Clearwater was selected as the one to release out of here, largely because of its shorter plant height, reduced lodging, and it did have the best yield out of these as well. So we really favored this lodging because we wanted a trait that would, or a variety that would be favorable for growers. If it's laying down flat, it's not going to be good for the growers. It's not going to be good for anyone. So we really put a lot of emphasis on that trait, somewhat to sacrificing seed size. Here's MN1501, that higher seed size variety. So that's something we may see in future varieties, is a larger seed size. So as soon as you release a variety, everybody wants to know what's coming next. And that's a very important question, because this being a perennial, growers that do have seed are already looking forward and thinking, well, do I want to plant more acreage of Clearwater? Or if there's something that's going to be markedly better coming along, should I wait and plant the new variety? So here's our pipeline. So MN Clearwater was initiated in 2015. So that was the first year that the SIN 1 generation was harvested. That's how we name these varieties. You can see that in 2016, 8 new ones were initiated. 2 are still in play. We have not done large seed increases on these, however, and then each year we establish a few more. So these new variety candidates are being developed, focusing on grain yield, threshability, shattering, seed size, and the next release is really 2022 at the earliest, and that's because we would have to initiate seed increase on these MN16 lines. So a couple years away yet. So here's same acknowledgements. I believe that Prabin had quite a number of folks involved here. But with that, I'll end and take questions if we have time.
Hey, thanks a lot, Jim. Good presentation. We have less than a minute for questions. If someone wants to shoot one in here at the bottom of your screen, send in a question. I'll wait for a few seconds here. If not, I'm sure Doug would be appreciative of one more minute. All right. Thanks, Jim. We'll move along to Doug Cattani. I'm pulling up Doug's information. All right. So this is Doug Cattani. He's worked with herbaceous perennials for 4 decades with emphasis on breeding, perennial grass plant development, and seed production. His current position is as the perennial crop breeder in the Department of Plant Sciences at the University of Manitoba, with the breeding of intermediate wheatgrass for grain production and perennial sunflower domestication as the major areas of interest. He's worked with industry, government, and academia at various times in his career in Canada and the United States. And today he's going to talk about his breeding work on intermediate wheatgrass at the University of Manitoba. Thanks, Doug. Take it away. Your slides are up. They look good.
Okay. Thank you, Lee.
We can hear you.
Good. As Lee said, I'm going to talk about breeding, but I'm going to tie it into agronomy because I think what we've heard across the talks today are the correlations from space planted to row planted or production type of growth environments. Some of our current breeding studies, I'm not going to throw a lot of data at you today. Just going to tell you what we're looking at and what we're trying to find out. These have all been mentioned by other speakers this morning, so none of it will be that new to you. We've made reciprocal crosses using 2 good yielding clones as one parent and a poor yielding one as the other parent or the reciprocal parent in each of those crosses. We've also chose parents that had different fusarium responses to inoculation in the field. So we hope to also be able to get at some of that. And the first harvests will be in 2020. So we will be just starting this work. And we're at 2 sites, Winnipeg, which is on a heavy clay loam, Red River Valley type of soil, and Red River Valley of the North, for those of you who may be south of Minnesota. And Kerman, which is a sandy clay loam, which is actually a beautiful soil for growing Kernza. The image on the right is the nursery. Each of the plants have been flagged. We've managed to overwinter them after last year's strange winter. And we'll monitor for seed yield and yield components for 3 years. All of our selections are done on 3 consecutive harvests for what we hope is continued seed yield for the producers in the fields. Jim had just mentioned that maybe we're looking at a different type of plant in production fields versus selection nurseries. And I'll talk a little bit about that as we go further. We are going to GBS one of these crosses, and analysis is going to be done with Sean Asselin. Sean is my former PhD student who's now working for Agriculture and Agri-Food Canada in Swift Current, Saskatchewan. Fusarium work will include field inoculation, and I'll be sharing a student, at least that's our current plan, with Matt Backer, who's now at the University of Manitoba, and as most of you know, is very, very knowledgeable on Fusarium. Okay, the other study that we've had ongoing actually now for 5 years is spaced versus row planted performance. We put out 2 space planted polycross, one at each of 2 sites. And we've replicated each clone 8 times within the site. We've planted progeny row within each site of each of the progeny from each polycross. So we can look at these on the site where we are evaluating the parent plus at a different site. And ultimately what we want to find out is what characters can we select for in nurseries for performance in the competitive growth environment or the field environment. One thing we've been looking at over the last number of years is that with our space plant plants, we measure the base of the plant or the area covered by each individual clone, because this does differ quite a bit between individuals, and then do a lot of our analysis based on a per area of ground area covered by the individual clones. We're hoping that may be able to give us a better indication of how they'll perform in a competitive growth environment. And having said that, I was out walking my progeny rows last week. And if you notice on the left, you can still see, and this is into their 5th production year, hopefully you can still see the individual rows in which this group was planted versus the image on the right where it looks like it's formed a fairly uniform sod. We're going to follow different this row planted still visible after 5 years, and a number that have no visible rows or very hard to distinguish the actual rows after 5 years, and do a comparison between them. And we'll look at seed yield, heads per unit area, yield per unit area as well on these. Jim gave a good talk on their cultivar development. We are also hopefully planting pre-breeder seed this week. We have had some rain today, which was sort of needed, and we want to get this in the field as soon as possible, but we may have to wait. We did have a previous candidate synthetic, and we ended up having to remove a clone, and what happened is it was throwing off a bluish purple seed, and through the seed sorter, which was color, we were removing about 15% of our yield. So the color of the clone was removed, the clone with the color was removed. Okay, I'm going to talk a little bit about our agronomic studies here, in part because a lot of what we do in breeding has to translate into agronomics. I gave an example on that previous slide of yield over time. This is a study that's being done that is going to look at the agronomics of intermediate wheatgrass seed production, animal use of the regrowth after harvest. And tomorrow when I talk about the phenology study, this will hopefully make a little more sense. We're also going to look at carbon and nitrogen cycling underneath these intermediate wheatgrass plots. And we're also throwing in a relatively non-aggressive legume, Alsike clover, interseeded between rows of intermediate wheatgrass, and not fertilizing those plots and looking at the benefits of having an intermediate wheat Alsike clover mixture. We did try to seed it last year. We were into our second year of a drought. This is a relatively sandy soil. We ended up with 1 or 2 seedlings per acre. That includes our forage control, which was alfalfa, and also tall fescue was in that mix. We just didn't get any germination from at this plot. It was the same seed lot used to seed all of our plots across Manitoba and Saskatchewan. And we've reseeded in August to make sure the seed was still alive, and it germinated and everything grew. So we're not sure what happened at this site, maybe just 2 years of bad drought on a fairly light soil. We have reseeded again. We finished Saturday at noon. We have an MSc student working on this project, Patrick Lehaget. He will be managing or looking into the agronomic aspects of intermediate wheatgrass production. We'll also have a graduate student in animal science looking at animal utilization of the fall aftermath. A graduate student working in agribusiness looking at the cost of production, predominantly of this, and the profitability. Martin Entz, an agronomist in our department at the University of Manitoba, is looking at intermediate wheatgrass in crop rotations. They did a fall seeding last year, and it is actually the plot on the left. The plot on the right that you can see a little better, it was fall rye. And this will remain in his rotation for 3 grain harvests. And he's looking at the benefits of the in rotation for the intermediate wheatgrass, and he may have a PhD student working on this in the near future. Okay, I'm going to talk about our current production guidelines because depending where you are, you will see different recommendations. Our recommendation in Manitoba is for spring seeding. The reason being is we have a lot of perennial weeds, and by spring seeding, we get mainly flushes of annual weeds with our intermediate. Our intermediate is able to compete relatively well with the annual weeds. We use a 12-inch row spacing or a 30 centimeter, depending which you like. We use a 6 pound per acre seeding rate. We want large individual plants as opposed to a solid string of smaller plants. In our organic production, we mow for weed control. First mowing is generally timed with flowering or just after flowering of our annual weeds to control those and also reduce seed rain back into the area. Now we're also looking into studies of underseeding the intermediate to wheat. And this is primarily to provide income to the producer in the first year as we do spring seeding. We did it, started last year on this where we have a conventional versus organic system. In the conventional system, we do use pesticides if needed. Where we have underseeded to wheat, we used herbicides that are registered for wheat use. And in our organic system, obviously, we couldn't use that. So weed control was strictly mowing after we took off the wheat crop. Seeding times were early spring and late spring, which in Manitoba hopefully equates to early May and late May, although this year, I'm not sure, even though we got it in early May, we had very little growing weather until late May. And we do an early fall and late fall seeding, much like Martin, I showed on Martin's study there. And the two different sites are the Red River clay in the top right, top left corner here. I'll get my hands straight. And you can see these were both taken a day apart this spring. You can barely see the rows of intermediate wheat that were underseeded to wheat last year. At Carmen, you can easily see the rows of intermediate wheatgrass, and I would expect this to show that the heavier clays are not as amenable to intermediate wheatgrass seed production as the lighter soils, the sandy loam on the bottom right versus the Montmorillonite type clay in the top image. What we're trying to figure out is how intermediate wheatgrass grows in Manitoba. We need to understand the developmental patterns of the plants and communities in order to be able to make selections for our growth environment. If you remember Steve's first graph of his, Steve Larson's first graph of his PCA analysis, Manitoba was shown to be basically on its own for the first two principal components. We are a different growth environment. We may be closer to northern Minnesota, but we still are different than many of the areas, and in part that is because we get strong winters and relatively little moisture. I've shown this image before, the annual growth cycle of a herbaceous perennial temperate grass. And yes, they have an annual growth cycle. You can substitute seeding for harvest if you want to start with the individual year, and then you take it through tiller growth and development. We get our reproductive tiller induction or vernalization. We also have to wait for a second stimuli for reproductive tiller development. Again, I will talk about that tomorrow with the phenology study. We then go through heading, flowering, pollination, and once we have fertilization, seed development, maturation, and then to harvest. Stresses during any of these periods can affect yield and also can affect the expression of the other seed yield components. So what have we found for keys for continued production? Fall regrowth in Manitoba is very important. We have a relatively short fall compared to all of the other growth areas that we have. Heat is important, but moisture may have a greater impact. And it's year dependent because we can't, at least under dry land production, no irrigation, we can't control moisture. And moisture seems to be a limiting factor. These are 2017 production or up to production. So harvest in 2016, the blue line follows to harvest in 17. The blue arrow actually shows when we cease to have good growing weather or daytime temperatures were below zero. And in 2017, winter came early and with very little moisture. So we almost had double the amount of moisture heading in. And if we look at the Winnipeg harvest, and that was Winnipeg, we also look at fertilizer timing in the fall application or spring for yield in 2017, it really made no difference. If you remember, we had this nice, long, relatively wet fall. Number of heads per row were almost exactly the same. Yields were a little different, at the two sites. And in 2018, which was the yellow line on the previous graph, you'll notice that even where we fertilize in fall, our yields drop precipitously, and that we figure is due to the lack of moisture. And 17, 18 were the first 2 years of our drought in Manitoba. I think in 16, the fall of 16, with that regrowth, the moisture from that year carried it through through harvest. If we look at the number of heads per meter of row, and these are actually on density related as well, we had double where we had fall fertility. So we need to push for tiller regrowth in the fall, better regrowth from our plants in order to maintain a yield. Whether it's a profitable yield in this year is debatable, but we had essentially double the number of seed heads and doubled the yield. When we looked between these two for yield per unit head, we saw about a 5 to 10% increase in the spring nitrogen over the fall applied nitrogen, showing that there wasn't a heck of a lot of compensation with that additional nitrogen in the spring, and that fall application pays off more often than not. We're also looking at timing of nutrition for continued seed yield post-harvest, spring, and then we're also doing a split to try and understand if we need as much in the fall or is a bit in the spring going to give us a boost. That's it for today. Thank you. With the time off, or I shouldn't say time off, but the time spent at home, I've been taking a lot of picture of birds through my picture window. And now that we've been allowed back out in the field, this mating pair of eagles sits and watches us on our blocks at work.
Great, Doug. Thank you for that presentation. We do have a minute for questions. I've got one for you to start with. The fall nitrogen application you just talked about, what was the timing on that?
Our fall is different from your fall.
Right, so when was it?
We try to get in before September 1, and we try to time it with at least a forecasted precipitation event.
So sometime in August.
Late August, yeah. We figure we probably have 6 to 8 weeks of good growth weather in a normal year. Last year was abnormal. We had a foot of snow on October 11th, which we'll find out what the impact of that on seed yield will be this year.
We have another question here from Anna. Could you tell us more about the origin and pedigree of your first Manitoba synthetic line that soon to become a variety?
The original clones came from your program, and we have a couple from the Grin accession as well, but this will be predominantly from the Land Institute materials we received. We made our selections, we've interbred them, and that's where we're at this point in time.
How many parents would be in this?
This will be a 9-clone synthetic. We made sure we did not take any 2 parents that came from the same line in your program. Mm-hmm. So we've tried to keep our diversity up pretty high.
Great. Excellent. Thank you, Doug, for that presentation, which actually concludes our presentation for this morning. I thank all of our speakers for today. I hope that each of them, if they haven't yet given us permission, Tessa Peters has sent you an invitation to approve the recording and dissemination of your talk. If you're cool with that, please fill that out, and hopefully we can get these posted soon to people who want to still watch them that couldn't make it this morning. We'll be back again tomorrow morning at 8 o'clock Central Daylight Time here in the US. Tessa, do you have anything else that you want to say here today?
No, I think that does it. Thanks everyone for attending and for participating, and I look forward to hearing what we have for you over the next week.
Yes, Tessa, thank you for making it so smooth. Worked very well. See you all tomorrow morning, I hope. Bye.
Well,